On behalf of everyone, welcome to a joint webinar sponsored by Picarro and EOSense. Today's webinar is going to be, you're going to be delighted by three speakers. Myself, my name is Gregor Lucic. I am a senior product manager at Picarro. I am looking after Picarro's greenhouse gas and trace gas line of analyzers and products. And I've been here since, I've been working for Picarro for the past 6 years. Joining me from EOSense is my colleague Deirdre Mallion. She got her MSc from the University of Alberta. She joined the EOSense team in 2016. And she's worked in market research, business development, as well as marketing. So Deirdre, thanks for being here. And I'd also like to introduce our speaker, Dr. Eri Saikawa. Eri is an associate professor and director of graduate studies at Emory University in Atlanta, Georgia. Her research covers a broad range of topics, including atmospheric chemistry, environmental health, biochemistry, climate science, and environmental policy. She's interested in understanding the sources and magnitudes of emissions linked to air pollution, ozone depletion, and climate change, and the impacts of these emissions on humans and society. She's also interested in what policy measures are available to reduce these emissions and how politics play a role in the policy-making process. So all of you, welcome, as well as our attendees. Thank you for being here. Just one last item of housekeeping before we begin. The webinar is being recorded, so you'll be able to rewatch it at a later date as well. The Zoom webinar has a Q&A function. So if you have any questions that come up during any one of our presentations, please go ahead and ask those questions in the Q&A function. We will get to them, and as many of them in the end. And for those that we can't get to, we will follow up after the webinar. With that, I'm going to transfer my control to Eri, and she will kick us off with her presentation.
Thank you so much. And hello, everybody. Good afternoon, or good morning, or good evening, depending on where you are. So great to have you all. And I wanted to thank Gregor, Deirdre, and Chris, and all the Picarro and EOSense team to have me on this webinar. So I wanted to start off with this picture. This is showing how agricultural emissions affect our health. And I'm taking this infographic by the European Environmental Bureau, as you see, because I think this infographic showcases how methane and ammonia that you're seeing in this picture are emitted from agriculture and how they create air pollution in the areas away from the agricultural areas. So methane is a greenhouse gas, and we're going to talk quite a bit about that. But it is also a precursor to ozone. I'm not sure if you can see it here. And tropospheric ozone is bad for your health, unlike the stratospheric ozone that protects us from the UV radiation. And ozone also impacts the ecosystems. And ammonia, on the other hand, leads to particulate matter, sometimes called as aerosols. And you might have heard PM, particulate matter for short. And we often call PM 2.5 for fine aerosols and PM 10 for coarse aerosols. These numbers are quite important because the smaller the size of the particles, they can get deeper into your lungs and cause respiratory and cardiovascular issues. And so that's why you're seeing here how ozone and PM can cause problems for your health. And I want to go into how agriculture is also related to climate change. So as you can see here, there are different sources that contribute to climate change. And the major part is electricity and heat, as you see here, 25%. But the second largest is agriculture, forestry, and land use. So you can see that when we think about agriculture, we have to think both about how it impacts air pollution and also climate change. Agriculture itself emits greenhouse gases, and the conversion of the land to agriculture from forestry also is a very important source for climate change. And I think we've all heard a lot about increasing the carbon sequestration potential for agriculture as a mitigation option for climate change. But I want to introduce you to other options in this webinar as well, knowing that agriculture is an important component contributing to climate change. So this figure is the most recent coming from the IPCC AR6, as you're seeing here. That stands for Intergovernmental Panel on Climate Change 6th Assessment Report. And so this is showing on the left-hand side here is the observed warming relative to the pre-industrial times. And we are seeing over 1 degree Celsius right now. And in the middle, it's showing the total human influence of this observed warming. So as you can see here, the natural component is very small, it's very minimal, but the total human influence is almost equal to what we are observing in the warming. You might also notice that the well-mixed greenhouse gases are actually contributing more, but because we have other human drivers that are cooling the atmosphere, this is the total human influence. And what is causing these total human influence is what is showing here. There are different gases, the main greenhouse gases, as we all know, carbon dioxide, methane, and nitrous oxide, as well as halogenated gases. And we talked about ammonia. Ammonia is actually cooling a little bit to the atmosphere. And organic carbon, sulfur dioxide, they also contribute to cooling of the atmosphere. But you also notice that the air bars are quite large. So these are very important components. There are many things that we don't understand. And some of them is because of aerosol formation and how aerosols actually lead to cloud formation, which is the biggest uncertainties for climate change right now. So I'm talking a lot about the atmospheric processes, and that's mainly because, as you've heard, my part of the work is really on atmospheric chemistry, and I wanted to give a brief background on how these emissions are very important for what we're talking about today. And so we just discussed the greenhouse gas emissions and how carbon dioxide, methane, and nitrous oxide are the biggest contributor to climate change in terms of warming the atmosphere. And we also discussed that methane and ammonia lead to air quality or air pollution. Here, what I'm showing is the sources of greenhouse gases, gas emissions in agriculture. And you can see that all the three major greenhouse gases that we talked about, carbon dioxide, methane, and nitrous oxide, they're all emitted in agriculture. So as you can see, CO2, they are emitted directly from the soil, but then they also go into accumulate soil carbon as well. So it's not just emissions. And so that is what we would be talking about today, how we need to think about the carbon and the nitrogen all together when we are trying to mitigate climate change issues. We talked about the forestry conversion to agricultural lands as well. So the harvesting also leads to this carbon cycle. And there is also fire that happened quite a bit in agricultural lands but to potentially manage the land. And also there are forest fires that take place. And in the West Coast, we are seeing quite bad wildfires that happen every year now. And methane coming from cows, as you were seeing in the earlier slides, and manure leading to both methane and nitrous oxide emissions. And from the soil, there is respiration of CO2. And also in rice fields, we see a lot of methane being emitted. I also want to mention, we will talk about how soils are sink for methane as well later on. And so we are finding a lot of different emissions coming from the soil, but also soils serving as a sink for these gases as well. The takeaway message from this is that agriculture is related to all these important greenhouse gas emissions and air pollutant emissions. And we really want to think about all of these emissions together, not just carbon. So options to mitigate climate change, I think this is a very important figure that was also published in the IPCC AR6 report. I'm sorry, I forgot to put the citation. But as you can see, there are different sectors. So you're seeing here, sorry, I'm trying to put the cursor. So you're seeing here different sectors like energy and buildings, transport, etc. And then here I'm highlighting the agriculture, forestry, and other land use over here. So what this figure is showing is the mitigation potential from different sectors. And you can clearly see that there is a very big potential to reduce emissions, the greenhouse gas emissions from agriculture. This cutter is related to cost. So you might find that some of the solutions are quite costly. But when you look into different solutions, I think the magnitude that is possible is something that we really need to think about. So carbon sequestration in agriculture that I just touched on a little bit has a very huge potential that's very similar to wind or solar energy, as you're seeing here. The cost is definitely higher, but there is also a very important potential for reducing the conversion of forests and other ecosystems as well. I think what we don't usually talk about much and that I want to dive into today is the potential for reducing methane and nitrous oxide emissions in agriculture, which is listed here. So those are pretty similar to the magnitude in other sectors, and the cost is not too high either. So can we think about this option, because we really need to do all of these above options to be able to get to where we would like. So in the past, my main interest was really on estimating emissions using the atmospheric data. So what I tried to do here is first, I tried to create the model for natural soil nitrous oxide emissions. I was very much interested in nitrous oxide. And then once I had that emissions estimates, I was able to collate other source emissions like agricultural soil emissions, biomass burning emissions, ocean emissions, and industry emissions. And then using the atmospheric observational data to optimize where these emissions might be coming from using a model. So what I'm showing you here is in these different graphs, this is the European total. This is for a different region total. The blue is what I had as the best guess. And then the different colors are showing the optimized emissions. And over here is the 2008 emissions total for different regions. And here is for each of the sectors. So you can see how agricultural soil is 21% of nitrous oxide emissions, and natural soil is about 40% of the global nitrous oxide emissions. So the soils are serving as a very important emitter of nitrous oxide. And you can see that Southern Asia is the biggest region where that's emitted. It might be a little bit too small, but Southern Asia is here. It actually includes South Asia and also most of the East and Northeast Asia, including China and Japan. And what was most interesting to us was that when we looked at this optimized emissions, Southern Asia agricultural soil was the only region and sector where we found significant increase in nitrous oxide emissions over time. Why is that? I think we all know that global nitrogen consumption has been contributing to nitrous oxide emissions. And this is showing the global nitrogen consumption over time. And what's really noticeable is this is the East Asia global nitrogen consumption. It has decreased a little bit, but until the 2008 point that I was talking about, it was increasing very substantially. And you can see here, this is South Asia increasing very steadily too. So we believe that this has contributed to the increase in nitrous oxide emissions from agriculture. So if that's the case, how can we reduce nitrous oxide emissions? And one of the ways that we were interested in was if we could use the living mulch system as shown here as a way to reduce the synthetic nitrogen fertilizer use and also reduce nitrous oxide emissions coming from the agricultural soil. So this is the photo taken by Dr. Nick Hill at UGA. So he's pioneered this system. What he did was he first plants white clover, which is a perennial cover crop, after the harvest is done over the winter. Because the winter is very mild in Georgia, they never die. And you can plant corn in the strip of white clover. The clover doesn't compete with corn or maize. And so then it actually is still alive even after the harvest. And because it's perennial, they can come back. And because it's a nitrogen fixer, it provides nitrogen to the soil, and that allows us to not need to put so much synthetic fertilizer to the corn or maize anymore. And I forgot to mention, but maize is the largest user of synthetic fertilizer, nitrogen fertilizer, and that's why we focused on corn. And so we tried to look at 4 different agricultural practices for growing corn or maize. And one was the bare soil with no cover crop. That's most popular in Georgia. And this is the living mulch system that I talked about. And we compare them to 2 different cover crops. Cereal rye, this is just for carbon because it is not a nitrogen fixer. Crimson clover is an annual nitrogen fixer cover crop. And we wanted to see if that makes any difference. So now moving on to the field measurements that you were most excited about. Sorry, I took a little longer in the introduction. But so what we started doing was to measure greenhouse gas emissions from the cornfield. In 2016 and 2017, we didn't have Picarro yet, so we did the GC measurement. But in 2018, we were lucky to be able to get the Picarro G2508. And so we created a setup like this in the cornfield in Georgia at the University of Georgia Agricultural Experimental Farm. And we were able to measure 6 plots at the same time, as you see here. And in the plot, we have a chamber like this, and we cap for an hour, and we measure 1 minute per chamber, 1 minute filter, and then go over the 6 chambers for an hour so that we can calculate the accumulation of gases over time and calculate fluxes. So just to show you the differences in soil health as well, before I move into the results for greenhouse gas fluxes and ammonia fluxes, I think it's very fascinating to see how the treatments are actually showing pretty big differences in soil health. So the no cover bare soil and cereal rye, we had to add 250 kilograms of nitrogen per hectare because there is no nitrogen source. Crimson clover and living mulch are both nitrogen fixers, and so we are adding fewer. These are based on the previous results, and this is the work by Dr. Hill. But what we can find is that the bulk density has substantially is significantly reduced for living mulch, as you can probably see here. And so that's also visible in the porosity. The KSAT has also significantly improved, so the water is flowing much faster in the living mulch. And as we would expect, the soil texture is not changing, so that we are not seeing any differences in the water held capacity. But you might notice the color of this living mulch soil, and that is having so much more labile carbon over time compared to the others. So now I want to move on to the soil CO2 fluxes. What I'm showing you here is the 2018 data where we were comparing the 4 different agricultural techniques that I talked about. So this TR is traditional bare soil. This red is the living mulch. orange is cereal rye, and green is crimson clover. These arrows, these black arrows are the times when we put the fertilizer, and these blue are when we had irrigation. So you can see at the beginning of the season, we are having much higher soil CO2 fluxes coming from the living mulch system. Over time, there aren't too many differences, as you see. And for soil nitrous oxide, as we would expect, after the fertilization, we are seeing much higher emissions. And as you would remember, cereal rye and traditional had much higher fertilizer put into these plots. And you might notice that we are having a higher living mulch emissions, especially when the clover, white clover, is dying. But at the end of the season, there's almost no emissions. This is the methane fluxes. And you know, I talked about soil serving as a sink, and this is what we are seeing. So this is the negative flux, and we are seeing soils to serve as the sink for methane, especially at the beginning of the season, and you can see also at the end of the season as well. There aren't too many differences among the plots, but you might notice that the traditional or the bare soil, these soils tend to serve as less of a methane sink than the cover crop soils. So what we did based off of these was to calculate the net carbon equivalence. We took into account 3 different greenhouse gas fluxes, soil carbon, and also I forgot to mention, but the fertilizer and herbicide application are quite different. If we use cover crop, we don't need to use as much fertilizer or herbicide compared to the bare soil. And so when we took those into account, the living mulch didn't seem to be small compared to the other practices, but this was because we didn't take into account the clover respiration. So when we also added, or rather subtracted the clover respiration component, the living mulch turned out to be the lowest in terms of the net carbon equivalence. It was even showing -16 to 60 kilograms per hectare per year. And so this is something that we are very interested in if this practice could serve as a way to reduce all the greenhouse gases and still increase soil carbon. And to do that, we were very fortunate to be able to borrow the automated chambers from EOSense last year. And so we started doing the field measurements using automated chambers as well as the manual chambers to hopefully quantify these emissions better. So you might remember the chamber setup. Now we have 6 chambers connected in a different way. And these are the EOSense automated chambers that we were able to use. And this is the one for manual chambers. And so what we did was so this is a different, slightly different design for 2021 funded by Syngenta. So what we focused on was 3 different agricultural practices. So this is the conventional bare soil, this is cereal rye, and this is intercrop with corn with soybeans, maize with soybeans. But we were also interested in the differences of the active ingredients like pesticides and herbicides. So we had no, low, high active ingredients for each of the agricultural practices. So, and then we had 4 replicates for these 9 practices and the level of active ingredients. What I'm showing you here as R1, R2 is how we measured. So we would usually start off with R6 and do the manual measurements first with these circles. These are the manual chambers. And then we would go through 6, 5, 4, 1, 2, 3, and then we would go back to 6 and then measure all of these with the automated chambers. Sorry, I actually misspoke. We started with the automated chambers for 36 hours, and then we continued with the manual chambers from 6 through 6, 5, 4, 1, 2, 3. I hope that makes sense. And this is important for what I'm going to talk about. So now I'm comparing the automated measurements and the manual measurements next to each other. This one is for CO2. You can see this is 2021 June through 2021 September. And you see much fewer data for manual measurements as you would expect, because we're only doing once per week. Here we're doing 36-hour measurements, and every time the chamber gets the data, we are putting that in. So for CO2, we're not seeing a very big difference over time. I'm comparing the CRI and the intercrop plot just because they both received fertilizer directly onto the plot, and that's easier to compare. But you can see that sometimes we have a very big outlier in the manual measurements that we are not seeing in the automated measurement. N2O was very interesting for us. So you can see right after fertilization, we are seeing a very big burst of nitrous oxide, as we would expect. And we are seeing some of that in the manual measurements, but as I'm going to show you later, it is quite different from what you're seeing from the automated. And you can see that there are some bursts, but then they start to go down, as I was showing you in 2018. So the nitrous oxide automated measurements can really show how it changes over the time of the 36 hours that we are measuring. And so this is shown in UTC. And so you would have to subtract 4 hours to get to the local time. And so this is about, I'm sorry, this is 12 o'clock. And so this is about 8 a.m. that we start measuring in the morning. And then you can see this increases during the day. And so what happened was when we did the manual measurements right after we finished the automated measurements, this is what we got. And so CRI, we're seeing a very big difference from what we were seeing in the automated measurements in the morning. And for intercrop, it was about what we might expect. So what I'm trying to showcase in this is that we learned a lot, that we really need to understand what time we need to do the manual measurements to be able to get the daily average correct. And there have been a lot of studies on this already. And it really just showcases how difficult it is to really get the correct, or rather general daily average, especially after the fertilization period when we are having such a big burst of nitrous oxide. So if we were to just have these data points from the manual measurements, we would potentially really either very much overestimate or underestimate, and this highlights the importance of automated measurements. This is methane. I think methane was quite interesting, but they had a pretty good, they shared a pretty good results between the automated and manual measurements. And you can see that the soils don't always serve as a sink, and sometimes it is emitting methane. And we see that in both automated and manual. And ammonia was the most challenging and most interesting for us, because in the automated measurements, we were quite able to capture right after fertilization. We were seeing a lot of bursts, first with ammonia, and then nitrous oxide later on after the irrigation. In the manual measurements, because we were doing it in so early in the morning, we were unable to capture those big bursts. So this also goes back to what I was talking about for the importance of how we measure. And there is a really big benefit to using automated chambers here. So when we look at different plots, as we have different plots in the manual measurements, we can see that depending on the time we measure, we are able to capture some of the bursts, but not as much as we can for the automated measurements. And I think this is very clear by looking at the automated measurements diurnal cycle, and together with soil moisture and soil temperature, the soil moisture was pretty different in the two plots as you see here. And this is the soil temperature. They go quite together. These pluses are the fluxes. And so again, this is 8 o'clock. So if we were to measure very early in the morning, we are missing these big peaks in the ammonia as well. And so this also highlights the importance again of when we measure. So just to summarize, I hope you took away that agriculture contributes both to air pollution and climate change. And I really believe that agriculture could be a very important solution to climate change mitigation, but we really need to think about how can we include all the greenhouse gases and ammonia together, not just thinking about climate change, but also air quality, because that's going to be important for our health and also for the ecosystem health as well. And climate smart agriculture solutions are getting a lot of highlight right now. And I think we need a bit more data for the southeastern US. We are really lacking that data, especially for corn and maize. It's true that they are mainly grown in the Midwest. I do think that the data from different regions is going to be very important for thinking about local-based solutions. And I hope I highlighted enough the importance of the measurements using automated chambers. And I think combined with Picarro, in situ really provided a very interesting data set. And I think having more of those would be very beneficial to understand how much is coming out of the agricultural soil. It is a bit of a challenge, and also thinking about ammonia is very difficult, but without the data, we cannot do modeling either. So I hope that more of that is going to come. So I just wanted to highlight some of my lab members. I'm here, but this is taken during the COVID period, but I have a lot of students that are very passionate about different projects. And I wanted to acknowledge different funding sources, especially Syngenta for funding this, and also USDA for the earlier work. And I would like to thank EOSense for letting us borrow the chambers. And these are the main folks in my lab, and these are the collaborators. Without them, we wouldn't have been able to do this work. So thank you very much, and I look forward to hearing your thoughts or comments, questions, and yes, I am done.
Thank you, Eri, so much for this wonderful presentation. Just as a reminder for our guests and attendees, we're going to be answering some questions at the end. We have some time at the end. So please put your questions in the Q&A section of the webinar. You are asking some questions into the chat, so we'll look after those as well. Next up is Deirdre, so I will hand off the screen to you.
Okay. There we go. Thanks, Gregor. I just wanted to start off really quickly by just telling everyone a little bit more about EOSense. So EOSense designs, develops, and manufactures hardware and software tools for measuring greenhouse gas emissions from soil. Our mission is to help researchers measure and visualize greenhouse gas emissions so they can better manage impacts of climate change. We were founded by a small team of soil flux scientists and engineers, and since then we've grown to a team of 13 people, and our office is located in Dartmouth, Nova Scotia, Canada. And EOSense offers a full suite of automated soil flux chamber systems. The EOS AC, which is shown here on the left, is the same type that was used by Eri for her study. These chambers are compact and they're designed to be used for long-term deployments, but they can also be used for shorter-term studies, and you can do survey measurements with them as well. Then in the middle here, we have the EOS ACLT and EOS ACLO chambers. The ACLT is the big transparent one. This one allows you to measure fluxes from more diverse types of vegetation, and it can also be used to measure net ecosystem exchange. The ACLO is the opaque version of the ACLT, and it's for use where dark measurements are preferred. Both of these chambers have an internal mixing fan as well as an internal auxiliary sensor port that can be used for measuring soil temperature and soil moisture inside of the chamber. And then in addition to our chambers, we also have two 12-port recirculating multiplexers. So all of EOSense's hardware is designed for long-term deployment and continuous data collection. This means researchers are better able to capture diurnal, seasonal, and longer-term trends in soil gas emissions like you saw in Eri's data. All of our hardware is fully integrated with Picarro analyzers. I've listed a few examples of compatible analyzers here, but the list is definitely not exhaustive. So if you do have a Picarro analyzer and you're interested in chambers, definitely get in touch with us. With our multiplexers, you can connect up to 12 chambers to a single analyzer. And we're also really excited to be releasing a new 16-port multiplexer in fall of 2022. One of the key benefits of the EOSense Picarro integration is the EOS Link AC software. This software runs directly on Picarro analyzers and allows users to fully customize and schedule chamber measurement cycles. So you can schedule and customize things like chamber open and closure durations, choosing the order in which the chambers are sampled, and customizing the gas line purge times between measurements. And because it runs directly on the analyzer, the software also automatically synchronizes the chamber closure timing with the analyzer timestamps. The data files from the analyzer can then be imported into our flux calculation software, which is called ES Analyze AC. And this software allows you to perform quick and easy visual curve fitting, and you can use both linear and exponential fits. Then you can export your flux data for further analysis in a variety of common file formats like CSV or XLS files. Yeah, I didn't want to talk for too long, so that's it for me. I'll pass it back over to Gregor, and he can tell you a little bit more about Picarro.
All right, thank you, Deirdre. I'm just going to take a minute or two to tell you a little bit about Picarro. Picarro, we provide solutions to measure greenhouse gas concentrations, trace gases, and stable isotopes across a lot of different scientific and research applications, along with providing solutions to the energy and utilities markets. There's about 250 plus employees here at Picarro, so I'm representing an absolutely amazing group of people that build, that market, and support the analyzers around the world. Today, there are thousands of Picarros that have been sold in over 95 countries all around the world. We sell some of these directly, or if your particular region is tailored to by partners, we also have a broad global network of partners around the world. We are an ISO 9001-2015 certified organization. Our headquarters is located here in Santa Clara, California, where we manufacture all of the Picarros and support them as well. Here are just a few pictures here of some of the team members that you've likely interacted with or will be interacting with if you're interested in our products. A picture of the laboratory here at headquarters where all the Picarros are built and tested, as well as just some pictures of our general headquarters office. Picarro sort of has its humble beginnings in the world of atmospheric monitoring. Some of the first applications that we tailored to were high precision continuous measurements of greenhouse gases in the atmosphere. And I'm just showing you a picture from perhaps one of the most famous stations in Hawaii, where we're continuously measuring the unfortunate increase in greenhouse gases in our atmosphere. But over the years, as our products have evolved and matured, the research community has challenged us with their many different and interesting applications. I've just selected a little bit of a subset of products and applications here that we've been tailoring to over the past few years. So you can see some colleagues here measuring greenhouse gas emissions in Antarctica using one of our portable instruments. You can see some colleagues from Europe that are performing continuous measurements of greenhouse gases and ammonia from a different type of agricultural emission source, so from feedlots and barns. And one of the things that's unique about our technology, and Eri spoke to this a little bit, is that it's mobile, it's portable, and it has seen deployments in all sorts of vehicles, from donkeys to airplanes to boats to small carts, large vehicles. And we're making greenhouse gas measurements and trace gas measurements on the go. Overall, our systems and solutions give you the ability to measure and monitor and quantify a variety of different compounds. We are focused in part on greenhouse gases and isotopes from some of these greenhouse gases: carbon dioxide, methane, nitrous oxide, water vapor, and some respective isotopes from those compounds as well. We also have quite a big portfolio of products for trace gases, some of the gases that are a little bit more difficult to measure or present lower concentrations in the atmosphere, like carbon monoxide, ammonia, formaldehyde, and so on and so on. These are the compounds that our products tailor to, but I wanted to highlight in particular some of the products that may be relevant for the kind of work you see here and some of the other applications I've highlighted. Eri spoke about the G2508, which is our 5-species continuous analyzer for carbon dioxide, methane, nitrous oxide, ammonia, and water vapor. A very popular product is also the G2201. It's an isotopic analyzer for carbon dioxide and methane. If you're doing incubation studies, if you're looking at microbial respiration, that's a very popular instrument to go around. And then I also mentioned in one of the previous pictures that there are portable versions of our instruments. So if you wish to take a backpack out into the field and perform certain measurements. And a lot of these analyzers that you see here, including the backpack, can be paired with EOSense chambers in the field as well. And I want to take just one last moment to highlight one of our newer additions to our product portfolio. It's an evolution of the G2508. It's a brand new product, the G2509. It has been optimized for improved gas response. Ammonia is a gas that is sticky and a little bit tricky to measure. We've provided it with some extended measurement ranges for those high flux environments, but nevertheless, we are able to maintain the part per billion and part per trillion sensitivity in the measurements. If you have any questions about any of these products, any compounds, or any applications that you might be interested in, please reach out to us and we'll be happy to have that discussion. With that, we've reached sort of the formal end of the presentations. I would ask my colleagues, Eri and Deirdre, to sort of come back on video and unmute yourself. I have a few questions here that we'll be going through that you've provided during the presentations. We'll try to get through as many as we can. Keep them coming. If there are some that we cannot, we'll follow up after the webinar as well. All right, let's start with one of the first ones in the Q&A box. Eri, in one of your initial slides, I think it was slide maybe 5, 4, or 6, you talked a little bit about the data that was coming from the IPCC 2002 report about other human drivers. What are some other human drivers that report is referring to?
Yeah, thank you for that question. So what they were doing was to try to differentiate the greenhouse gases and other gases that contribute to to climate change. And so in the other human drivers, it's mainly the sulfur dioxide and also the nitrogen oxides. So they're not really considered greenhouse gases, but what they do is sulfur dioxide is important in forming sulfate aerosols that cool the atmosphere. And for nitrogen oxides, they actually produce hydroxyl radical that is an important loss mechanism for methane. And because of that, they act as cooling the atmosphere. And so that was the differentiation of the IPCC report. And I hope that's clear now.
Great, thank you. Eri, I hope you're ready. There's more questions here for you. Let's see how many we can get through. There's a question here: how much calculated nitrogen was retained from the clover?
Yeah, so I just posted the paper that we published on the in the frontiers of the environment as well. And so there is data on nitrogen too, but that is something that we really want to get into. And for example, my collaborators, Nick Hill and others have also been looking into that. So I'm not able to say on top of my head how much, but I hope the paper is helpful and we will hopefully have more answers to come in the future.
Excellent, and I'm sure we'll share some of the references to those publications after the webinar as well. I'm just going to tack on to this question, another one. I think some of the attendees have been wondering what aspects of your research have been published, and what are you still sort of looking forward to publishing?
Yeah, so that's a great question. So we've published the work that we did in 2016 and 2017 using the GC. And then we published the work in 2018 and 2019 that I talked about a little bit. I didn't go into the 2019 detail, but we are still working on the data from 2021. We are continuing that field work this year. And so hopefully we can get more data and then publish. In the future, we would love to also expand to different systems and then figure out what would be the best way to really measure using these different approaches.
There's a question here: did you have to apply any herbicide to kill the clover, or just strip till?
Yeah, so that's a great question too. So yes, we did have to kill the clover using the herbicide. And so what happens usually is that in the cereal rye plot, they use the herbicide everywhere to kill first. And then that's the same thing for crimson clover. That was the annual cover crop. But for the living mulch, it's only killing a strip of it. And so the amount of herbicide that's used is much less. And I should also mention that these are all no-till plots. Sorry.
Another question for you. How well does temperature correlate with diurnal changes in the flux of nitrous oxide and ammonia?
Yeah, so that's exactly what we are looking at right now. And so I showed you the sneak peek of that. And so we would like to understand what is the main driver for these emissions. I think what I was showing, I hope some of that was clear, maybe not, but I showed the ammonia flux. It seemed like the temperature wasn't really impacting the ammonia too much. But obviously the fertilizer, I think I'm going to another question a little bit. I think there was a question about fertilizer and ammonia. We are seeing a very big burst of ammonia first, right after the fertilization, and then nitrous oxide. And this is mainly because of the type of fertilizer. This is the liquid fertilizer that they are applying. And when that's urea more solid, then that's also different. So I think there are very many interesting questions to ask. How does the application of fertilizer impact both the nitrous oxide and ammonia, et cetera, as well?
Yeah, excellent. I think that covers the other question about the high ammonia fluxes. There have been a couple of questions coming in when you were sort of at the heart of your data presentation. And they were about, some of the differences between the static measurements, the manual sort of chamber measurements, and the automated measurements, the closure times, the frequency of collection, and things like that. So if you could just speak to that a little bit, some of the differences between closure times for the manual chambers, for the automated chambers.
Yeah, thank you for the questions in the chat. Sorry, that was not very clear. So maybe I will clarify. okay, so in 2018 and well, in all years, what we've been doing is to measure at least weekly. And so that was the case in 2016, 2017, 2018. We measured weekly with the manual measurements. In 2019, we did a very different study design where we measured daily after fertilization, focused on how it affects the emissions right after the first and the second fertilization. So if you're interested, you can look into that in the paper that I posted. In 2021, we were able to use the automated chambers. And so we did the weekly, but then what we would do is we would measure for 2 days in a week. So for the 36 hours, we would measure with the automated chambers, and then we would measure with the manual. This year, we are doing the same thing. And so even though we are saying automated, we cannot leave it open, working for the whole period. And that is mainly because we don't have a power source. And so we are using the diesel generator. Maybe you might have noticed in the picture that I showed. And so it is very difficult to continuously measure for the whole growing season. They do have to irrigate, etc. too. And so that's the limitation. The 2 days a week is the best we can do. But that's great to be able to use the automated chambers to at least have the diurnal cycle and how it changes over time. In terms of the plots, maybe it is easier if you could move the slides to, which slide number was that? Slide 20, I believe.
This one?
Yeah, thank you. So sorry, it was not very clear. What I wanted to say was, I'm trying to get to this slide. Okay. So, okay, so if you could give me the control so I can highlight, if that's okay.
Yeah.
Thank you so much.
So
we have four replicates in this plot. So first row here is having conventional no, conventional low, conventional high. These are all bare soil with 3 different active ingredient levels. These are all cereal rye, the 3 different active ingredient levels, and these are intercrop, so corn together with soybeans. So it's a little bit different from the living mulch, but we wanted to do something similar. And then we have the replicates. So this is the replicate 1, replicate 2, and replicate 3. So these are randomized. And these squares are showing how we measure the manual measurements. And so these circles are the manual chambers. So we have 1 chamber per plot, and this P is where Picarro is located. So we would put the tubings like this to measure 6 chambers at the same time. And we would first go with this one measuring for 1 minute, and then go to the filter for 1 minute, and this for 1 minute, for filter 1 minute, etc. And then we will go for the 6 chambers, 5 rounds. And that's how we calculate the flux for the manual measurement. So we do the 1 hour measurement 6 times in a day. And that's after the 36-hour measurements of these squares. So these squares are the EOSense chambers. And so we have 6 EOSense chambers covering the no high of each of the 3 techniques. And we measure with Picarro here. There was, I think, a question about how close the Picarro should be to the chambers. We try to be as close as possible for ammonia. We know that very long tubings can be, can cause issues for ammonia because they're sticky. So that's also something that we've been trying. And so that's why we're trying to be as close as possible and looking at if the length of the tubing matters or not. I hope that covered the questions. Was there anything else about the study design that I missed?
I think this is, I think you did a pretty good job at addressing quite a few comments. We're almost at the end, but I think we can squeeze in a couple of more questions. I think there's one here for Deirdre. Let me just swap over to a slide that might be helpful. There's a question that's wondering, is there a way to measure greenhouse gas emissions using the EOSense chambers without Picarro analyzing?
Yeah, so there's no sensor built into the chambers themselves, so you do need some manner of analyzer. We are compatible with other analyzers. So if you have an analyzer and you're not sure, you can get in touch with us, and we'll let you know if it's something we're compatible with.
Yeah, thank you. There's a question here for us, Picarro, which I think I can answer. I'll go to this slide just so it's helpful. Does the Picarro analyzer measure the concentration of the target gas using a chemical sensor or a laser? So the Picarro analyzer is based on a technology principle called cavity ring-down spectroscopy. It's a laser-based measurement. It's a continuous measurement. The system performs roughly 300 laser-based measurements per second, and it usually has a measurement response of about 1 measurement per second. And that can vary depending on the model and how many things it can measure.
Sorry, I should have mentioned that for EOSense chambers, we are capping for 10 minutes. And then we continue that for different chambers for 36 hours. I guess I didn't mention that.
Maybe one last question for you, Deirdre. You talked a little bit about the different types of chambers that you have. How much can people mix and match the various chambers for their applications?
Yeah, so if you have a multiplexer, you can put any of our chambers in there. So you could have any combination of chambers that you want, and all of them will work seamlessly with Picarro. So mix and match away.
Okay, perfect. I think that's it. We're almost out of time. Again, thank you, first of all, to the speakers and my colleagues at EOSense for helping with the presentation. Eri, it's been a pleasure hosting you. The presentation was absolutely lovely. We'll be following up after the presentation with information about the recording and some other questions that we didn't manage to answer. Thank you so much for attending. I hope you enjoyed it, and I wish you all a good day. Thank you again to the speakers.
Thank you so much.